We are group 3, so Tina, Aditi, Zoe, Lucy, and Tian, and Justin, and today we'll be presenting some novel treatments to Leber congenital amaurosis type 1. Okay. Leber's congenital amaurosis, or LCA, is a group of early onset autosomal, predominantly recessive dystrophies affecting the retina. It results from mutations in at least 22 different genes, with at least 17 different types currently described, each differentiated by their genetic cause and phenotypic variation in vision loss and eye abnormalities. Naturally, the genetic heterogeneity of its causes makes it difficult to both study and treat. LCA1, which results from mutations in the GUC-Y2D gene on chromosome 17, was the first type of LCA to be described and is also the most prevalent. So GUC-Y2D encodes for guanylate cyclase 1, or GC1, a transmembrane protein produced predominantly in the cones of the retina, which plays a key role in the phototransduction pathway. The phototransduction pathway is a complex biochemical cascade that occurs within the cone of the eye, commencing when a photon of light causes a conformational change in rhodopsin. This leads to the hydrolyzation of cyclic GMP, resulting in the closure of cyclic GMP-gated ion channels and consequently a reduction of intracellular calcium. This eventually leads to the hyperpolarization of the photoreceptor. GC1 plays a role in helping to return the receptor to its dark state by resynthesizing cyclic GMP from GTP. Hence, mutations in the GUC-Y2D gene can affect the efficacy of GC1 in fulfilling its function within this pathway, leading to the biochemical equivalent of chronic light exposure for your cells. Consequently, while many patients generally retain normal photoreceptor lamina architecture, the patients are routinely described as having profound visual impairment, such as reduced visual acuity and lack of color perception, which stems predominantly from their loss of cone function. LAC1 mutations are found all the way along GUC-Y2 gene, with pathogenic mutations still being reported, and the prevalence of certain mutations varying greatly depending on the cohort that's being studied. In general, though, they can be separated into two groups based on the functional impact on GC1. The first are frameshift mutations that destroy the enzymatic activity of the protein by truncating away the functionally important catalytic site, and then the second are missense mutations, which don't actually alter the protein size, and though they can have very varied consequences, ranging from seemingly null effects to complete inactivation of the enzymatic activity of the protein. Corrective lenses and visual aids is currently the main treatment for general LCA treatments. They increase mobility, independence, and improve quality of life for their users, but they are mere band-aids compared to gene therapy and a possible cure.
Although there are 18 variants of LCA, only a few gene therapies exist. The FDA has approved one treatment for LCA2 called Luxturna. It involves a single injection into viable retinal pigment epithelial cells and restores protein synthesis function by transducing the wild-type gene. It has shown significant improvements in all 15 patients. An LCA4 study on mice has shown effective gene replacement therapy in both rods and cones using adeno-associated virus AAV8 vector and human RK promoter, restoring photoreceptor enzyme synthesis. Current research into LCA8 has linked the CRB1 gene to retinal cell villi and consequential retinal integrity. There is a study investigating recombinant AAV vectors with small promoter regions to accommodate this large gene for gene substitution therapies. Another study demonstrated the complexity of CRB1 mutations with possible environmental factors altering phenotype expression. Meanwhile, there are two ongoing human trials for LCA10 treatments. EDIT-101 is a CRISPR-based method with non-homologous end joining delivered via AAV into the retina to substitute the correct base. And sepofarsen involves multiple in vivo injections of RNA-based antisense oligonucleotides therapy into the back of the eye to restore RNA splice functioning. LCA1, which we are researching, doesn't have any current genetic treatments. However, patients usually have preserved retinal appearance and only lack functioning retinal GC1 isoenzymes, so there is great potential for gene therapy. There is existing animal research, however. A study in mice using a human promoter and AAV8 vector restored rod and cone photoreceptor enzyme function that the human GUC-Y2D retinal gene codes for. And another demonstrated the functional use of the same promoter with AAV on primate photoreceptor cells. These methods could be applied to humans using CRISPR. But what is CRISPR? CRISPR technology refers to specialized gene editing tools often used to treat genetic diseases. The functional format varies a lot. Type 2 CRISPR-Cas systems, for example, mainly use the Cas9 protein complex with a single guide RNA programmed to target a specific DNA sequence. The guide RNA consists of tracer RNA that activates CRISPR RNA, which make the hairpin loop to stabilize the complex. The guide RNA is then led to the target DNA sequence by the PAM, protospacer adjacent motif. Once matched, a double-stranded break is made on the target strand by H and H and on the complementary strand by RUVC domains in the Cas9 enzyme. Deletion of nucleotides occurs and new DNA can be introduced. The double-stranded break is repaired via NAEJ or HDR, thus incorporating the edit into the genome, resulting in a loss or gain of function via random error or custom repair, respectively. Base editing can also occur using a less energetic Cas9 complex, which is a newer development. The target strand is nicked or the base is converted, for example, C to T, then the complementary strand is synthesized, so an A complements the T, and the new edit is replicated via normal DNA repair processes, so the A becomes the T. This results in an altered DNA sequence without cleavage. CRISPR complexes can be delivered in vitro, in vivo, and ex vivo by a plasmid, a viral vector, and RNP complex nanoparticles or other means. We are investigating two main CRISPR-based strategies to treat LCA1.
So as discussed previously, there are two main types of mutations for LCA1 and thus you have two main strategies. So for missense mutations on the left, we present a base editing C to T approach, which is a more traditional approach with greater evidence. As an extension for frameshift mutations on the right, we present a prime editing approach where we insert base C, but we believe this method has been less researched and still has a few major problems. So first up is base editing. So firstly, we consider the Phe999 SER mutation, which is a T to C point mutation at chromosome 17, exon 16, that occurs in approximately 0.1% of LCA1 patients. So we consider this to be a reasonably good model mutation to showcase some of the benefits and limitations of base editing, as techniques for converting C to T bases are reasonably well documented for other diseases. So biochemically, it changes a large phenylalanine molecule to a small polar serine molecule, and so not too much is known about this mutation, but it's thought to affect stability of the GC1 protein. So the general process of this approach involves attaching a cytidine deaminase enzyme to dCas9 and binding DNA to it. C is converted to U via deamination, while U is converted to T by using the BE3 variant of cytidine deaminase. This makes the edit more specific and reliable. So the main advantages over conventional methods, i.e. cutting out the gene via Cas9 and NHJ, are that there's very little off-target effects. This doesn't require a double-stranded DNA backbone cleavage or a donor template, meaning we don't have to cut out the entire backbone, which reduces the chance of random mutations. So here we describe the process in more detail. So first of all, we bind DNA to the dCas9 complex. So we have to make sgRNA with the original code for the gene you want to edit. In this case, a 20 nucleotide region approximately around the active region ACTC-TCC. Then we take our original genomic DNA, which will enter the dCas9 site DE complex, which opens up the two strands at the site defined by sgRNA. On the bottom 5' to 3' coding strand, C is converted to U via deamination by the cytidine deaminase enzyme, and the deamination is performed via the hydrolysis of cytosine into uracil. And then DNA replication methods via the BE3 variant of the cytidine deaminase enzyme are then used to convert the U permanently to T. So DNA replication repair mechanisms can either repair the unmodified 3' to 5' strand containing the complementary G at the top, or the modified 5' to 3' strand containing U at the bottom. And so the BE3 variant of the enzyme favors repair of the modified U strand compared to the existing G strand. So in other words, the 5' to 3' U strand is used as a template to repair the original strand, converting G to A via cellular mismatch repair by B3. Then standard DNA replication repair methods are then used to convert U to T by using the newly modified A strand as a template, and so we end up with the TTT sequence. So a little bit more about BE3. So BE3 is an efficient base editor favoring repair of the edited strand. More accurately, it recognizes Cs that immediately follows Ts, so a TC sequence. So for BE3 to be used, it needs to recognize an NGG PAM sequence roughly 15 bases downstream of the area you want to alter, and so the sequence alignment is shown on the screen. So the TC we want to edit is in the light green region, meaning that it's less likely to be edited, but the major problem is that we've got another TC in a dark green region right next to it, which means that it's more likely to be edited. And so BE3 is likely to edit all TC to TT within the entire green regions. The impact of changing the second C to a T is relatively unknown. It is possible they might not actually do anything.
The base editor is then expected to be delivered via recombinant adeno-associated virus, which has shown promising results in both mice and human treatments of LCA10. As previously addressed, some treatments are undergoing human trials, which involve certain combinations of subretinal injections of Cas9 complexes and guide RNAs via AAVs. There are a range of delivery methods as presented here. However, we would like to formally present the adeno-associated viral subretinal injection as the most likely to successfully be delivered in vivo and directly to photoreceptor cells whilst effectively carrying the complex. However, we propose the efficient packaging of the BE3 complex into dual AAV vectors in order to facilitate its 5 to 6 kilobases weight, as a single AAV can only carry up to 4.7 kilobases, with the aim of transforming at least 10% of photoreceptors in the individual. We expect to first attempt to deliver this solution into a genetically modified mouse with a mutated gene knocked in to the retina, which has shown success in previous trials. Therefore, base editing presents advantages. It may be a more reliable method as it doesn't require both DNA strands to break. Furthermore, it has a high editing efficiency of up to 98%, which lowers the probability of indels or random mutations, with an error rate of about 1% for adenine-based editors, which is only a little higher for cytosine-based editors. This ultimately minimizes off-target effects and does not require repair templates. However, the limitations of traditional base editing involve the limited capacity, as only conversions from C to T and A to G are currently possible. It doesn't address frame shift mutations, and furthermore, personalized DNA sequencing is required for treatment design, which is very costly. It could cause bystander edits, especially in adjacent TCs. Therefore, base editing may be reliable, but its accuracy is doubtful when dealing with missense mutations. However, this limitation would be addressed through experimentation on mice models. Because of these disadvantages, we propose prime editing as another approach to other mutations.
So as we previously mentioned, base editing is unable to correct frame shift mutations, which are often the most pathogenic mutations in this disease. Base editors are also unable to perform 8 of the 12 possible base transformations in gene editing. So for such a heterogeneous disease, we also need a strategy to deal with a broader scope of mutations. So here from a 2007 North American cohort study, we could identify a single frame shift mutation with a relatively high prevalence. This is the threonine 839DL1AC mutation, which is a deletion of a single cytosine base seen here. We will attempt to correct this mutation using a prime editing strategy. Prime editing is much less restricted by the proximity of PAM sequences, only needing at least one sequence within 30 base pairs downstream. So as you can see in this image, we have 3 PAM sequences that could potentially be used. In prior Crispr trials with LCA, they have aimed for a transformation of 10% of photoreceptor cells in the fovea, which was able to restore visual acuity. Achieving this is entirely dependent on our editing efficiency, which is largely based on the effectiveness of our delivery method. This study also found that editing efficiency is dose dependent, which is another consideration when we're choosing delivery methods. The prime editor differs from the base editor in a few key ways. The prime editor uses a weakened Cas9 enzyme called nickase that performs a single-stranded cut in the DNA. The complex also contains a reverse transcriptase domain to synthesize new DNA from our RNA template. The RNA template is known as prime editing guide RNA, or PEG RNA, and contains a binding region and the sequence of our edited gene, including the inserted cytosine base. We will use prime editor 3 as this was found to be the most efficient prime editor. This means we're also going to need a single guide RNA, but more on that in a second. So to insert our gene, the PEG RNA will guide the PE3 complex to our region of interest, where the nickase will cut the strand to be edited. The reverse transcriptase will then extend the DNA backbone of the 3' flap in the nick based on our edited sequence and inserting the cytosine. This leaves a newly edited strand and the complex will dissociate. So now we'll have 2 flaps of DNA on our edited strand. This is because the strands are now of unequal length and the nick allows them to undergo flap equilibration. This is where the 5' flap and the 3' flap will alternately bind and dissociate to and from the unedited strand. Eventually, the 5' flap will be cleaved by cellular machinery, and the 3' flap containing our edit will be retained. The edited strand will then be ligated. However, our inserted cytosine base will create a mismatch, and this will be recognized by the cell. The cell will then attempt to repair it, but we need to convince the cell to repair the new deletion mutation on the unedited strand rather than our new insertion mutation on the edited strand. This is where prime editor 3 is very effective. So prime editor 3 uses a single guide RNA to guide the complex to perform a second nick on the unedited strand. The sgRNA is complementary to our edited sequence, meaning that the second nick will not be performed until our edit has been incorporated. The second nick will trigger the cell to recognize the unedited strand as the mutant and rebuild it according to our edited sequence, successfully inserting the CG pair into the double-stranded DNA.
Right, so as we've mentioned before, for the base editors, there's so many viral and non-viral approaches to delivering the editing complex. And considering that we still want an in vivo treatment, adeno and lentiviral vectors, which will trigger unwanted side effects, might not be viable. The DNA-free vesicle delivery is much safer, but as we mentioned earlier, it has a really low editing efficiency in vivo at about 1 to 2%. The most promising method is again the adeno-associated delivery, safer and well researched. While the P3 complex can be much larger than the packaging capacity of AAV, we could overcome this using the same dual delivery method. However, because prime editing is a really recent development, its in vivo delivery is less well understood. While both strategies have advantages over the conventional therapies, they have problems. Embryo editing is illegal in most regions, and this whole gene editing concept is still fairly controversial, especially how it may change the population gene makeup. At the moment, the cost of Crispr treatment is high enough to stop most of the world's population from gaining any benefits. Also, because there are just so many different mutations causing LCA1, the treatment will need to be tailored to suit each individual, causing extra money. In terms of the technology itself, although we share the same target gene with the model organism, the mouse, the mutations are likely to be different and will eventually need human trials. Some people can be immune to the classic Staphylococcus-derived Cas9 factors, and there's always the issue with off-target edit and insertion of mutagenesis. And when something goes wrong in the eyes, we don't know how wrong it can get. Overall, both the base editing and prime editing strategies are theoretically viable and promising and really should provide a novel treatment to LCA1, which has been overlooked in the past. However, the real effect of these treatments will have to be determined in lab trials. And these are the references we used. Thank you for paying attention to our presentation. And any questions?